Our next presenter is Brad Rogers of GradienT. Brad, you may begin.
Good afternoon. My name is Dr. Brad Rogers, and I'm the lead scientist at GradienT Technologies. Today, I'm very excited to introduce you to TempiSure, our lab-on-a-chip platform for measuring drug stability. Unstable drugs are a nightmare for the pharmaceutical industry because they're difficult to manufacture and they have short shelf lives. TempiSure's small-scale design allows scientists to predict these problems early on when their drug materials are limited. Did you know it takes over $6 million in 5 years to bring a single drug into clinical trials? The research phase of this process involves generating small quantities of many drug candidates and then selecting a handful that are potent and safe. Then a single drug is sent on to development to be produced in large quantities for clinical trials. In order for a pharmaceutical company to outrace their competitors to market, it's critical to avoid delays and failures. Choosing the right drug candidate, however, is particularly challenging because the amount of material is very limited for accurate risk assessments. TempiSure is a superior platform for assessing stability risks, which has the potential to reduce development failures by 25%. Our patent-pending design exposes a small amount of precious drug material to a range of temperatures simultaneously. And videos are recorded to watch the drug precipitate over time. The industry standard test for predicting stability only works for low concentration drug products. And our closest competitor offers an instrument that can't measure stability over time, and it requires by hand sample preparation, unlike TempiSure. Our business plan is a simple razor and razor blades model. We offer a $100,000 benchtop instrument that uses $50 consumable sample chips. There's a significant need for this product in the biopharmaceutical space, where we expect $100 million in instrument sales and $40 million every year from the consumable chips. In fact, we've already raised over $1 million through collaborations with AstraZeneca and Sanofi to test this platform on their drugs. My team at Penn State and our biopharmaceutical collaborators are currently focused on prototype development. We plan to form a company and begin manufacturing in 2022. And by 2023, we will launch sales to dozens of biotechs across the world, thereby eliminating stability risks and accelerating times to market. Why settle for being sure that a product is stable when you can be TempiSure? Thank you for your time, and I'll be happy to take any questions.
Thank you, Brad. Nice presentation. We will now start with the Q&A, beginning with Alana. Alana, do you want to kick us off?
Sure, thank you. That was a great presentation, very interesting problem you're solving. Tell me a little bit about the manufacturing side. You mentioned that you need to form a manufacturing capability. Have you done some studies to determine that this is indeed manufacturable at scale, reliably, and at the price that you're quoting, you know, $100,000 cost? And that includes those sample chips as well.
Yeah, that's a great question. We're early on, so we're actually still developing the prototype, and we have the raw materials are about $10,000. So we're going to have to explore these questions you're mentioning. How does that actually translate to large scale manufacturing? And we're actually very interested in partnering with some existing instrument manufacturers to hopefully eliminate some of the costs on our side and leverage their existing relationships in this space.
Mm-hmm. Thank you.
Thank you for the question.
Brad, very nice presentation, and congratulations on raising $1 million already. That's quite an achievement at this stage of the game.
Thanks.
I'm a little bit curious about what did you do, or what are you doing with that cash, and can you speak a little bit about the results you've seen with whatever compounds AZ and Sanofi have sent you?
Yeah, so we've collaborated with these pharmaceutical companies for about 5 years now. The first projects with AZ were focused on formulation chemistry, and we published a paper in 2019 with them. And over time, that these projects kind of evolved to develop our instrumentation for predicting shelf life. And I'm really excited to share this, but last year in our collaboration with Sanofi, we did a blind study where they provided us with a handful of their drugs and they know the shelf life, and they asked, can our instrument predict that? And we got some really promising results that shows there's a strong correlation, and we're writing that up into a manuscript that will serve as our first major piece of advertisement.
How did they react to your result?
I think very, very excited. We actually had a meeting with them a month or two ago talking about potentially onboarding or taking our prototype for a test drive in their lab. That's what they see as the next step. They want to try it out for themselves.
Okay, thank you.
You're welcome. Thank you.
Hey, Brad, thanks for the presentation. So does your device only cover small molecules, or can you work with other like biologics, for example?
Yeah, good question. We've almost primarily focused on biologics. Monoclonal antibodies are the biggest biologic on the market right now, and that's what we've been focused on. It's an open question as to how big of a scope can this technology be applied to, but we've primarily focused on biologics up to this point.
Great. What's the temperature range that you can, I guess, your gradient covers, and what's the sensitivity if you're able to fine tune that?
Yeah, our hardware can go anywhere from -20 on the cold side, that's degrees Celsius, up to the boiling point of water, 100 degrees. So that's plenty of room for a biologic, but for other materials, we might have to explore different designs to go farther. As far as fine tuning goes, you're limited by sort of the microscope on spatially resolving these temperature gradients.
All right, thanks.
Thank you.
Thank you, judges, for your questions, and thank you, Brad, for your presentation.
Thank you for the opportunity.